Thank you so much. It's a huge pleasure to be here with you this morning. And I'm really excited to talk to you about assessing the value of new diagnostics. Might not be something that you think about every day, but with the proliferation of new diagnostics, which is really exciting for us in the clinical microbiology laboratory, as well as for patients and other healthcare practitioners, we really need to think about this. So the objectives of my presentation today are to help you understand the use of randomized clinical trials to estimate value of new diagnostics, to appreciate the value of guided results interpretation, and to describe test ordering guidance and ordering algorithms. And I'm going to show you some examples, some real life examples of what you might do in clinical practice. But first, I want to pause and talk for a minute about value, because I think when we say value, a lot of people might think we're just talking about money, but those of us who went into healthcare probably didn't go into healthcare for money. We went into healthcare to help patients. And I think value for us, for healthcare practitioners, is really about making patients' lives better, helping humanity. And so when we define value, we tend to think about patient-related outcomes, and I'll show you some of those. And then when we're thinking about the crisis that we have in antibacterial resistance, there's another value. And I would propose to you that that value is the appropriate use of antibiotics, because they are a precious resource that we need to preserve. But you'll see there are different ways of measuring value, and you can walk away and decide how you would apply value in terms of your clinical practice, or in this case, the use of diagnostic tests. So I'm going to talk first about randomized clinical trials. You might think, well, why would you do that? That seems extraordinarily complicated for a diagnostic test. What's typically done is to do what's called a pre and post intervention study. That is, you have a new diagnostic that comes along and you implement it in your clinical practice. And then after a period of time, you look at how your patients did when you were using it, and then how the patients did before you started using it. But the problem with that is there can be non-study variations that occur over time that can affect patient outcomes. Imagine, for example, that you implement a new diagnostic and then a COVID-19 outbreak happens. You can't really compare the time before that pandemic and afterwards, even though you might be trying to look at the value of a new diagnostic test. And there are many changes, of course, that come along in healthcare. So let me show you a couple of examples where we've executed randomized controlled clinical trials. And the first is looking at blood culture bottle panels. So I'm going to take you back almost a decade now to the year 2013, so 8 years ago, when the film array BCID panel was introduced into clinical practice by means of being cleared by the FDA in the United States, at least. And so prior to that, if you had a positive blood culture bottle, we in the laboratory, we would do a gram stain on it. We would issue a report rapidly of that gram stain that would say, in this case, gram-positive cocci resembling staphylococci. And then we would set up a subculture, and it would take us another day to get to an identification, and probably a day after that to get to susceptibility results. But as you know, we've had a proliferation of blood culture panels to test these positive blood cultures. And I know you can't read this, and that's okay, because this is showing you all the different bacterial species now that we can detect with the different panels that are available. This is in the US. You may have slightly different panels available where you are, but the point is the same. Lots of different gram-positive bacteria, gram-negative bacteria, Candida species, some filamentous fungi, other yeasts, and a plethora of resistance genes. And so when these panels first came out, we asked ourselves, are these valuable in our clinical practice? They are, after all, expensive. They are add-on tests because they don't replace a lot of what you already do. And you have to run them around the clock to realize value. That's complicated for any healthcare facility. And will clinicians actually change anything based on the results of these tests? They're pretty easy to run in the laboratory. This is an example of the film array blood culture identification panel, but as I showed you, there are other companies that have panels. They're all pretty easy to use. In this case, you load up a little pouch with the material from your positive blood culture bottle, and you put that pouch on the instrument, and in about an hour, you get all of those results. This is what loading the pouch actually looks like in the laboratory. So it's not too hard to do. This takes 1 to 2 minutes to do, but you have to be standing by for these, and you have to be ready to do this in real time. And as you can imagine, this is fairly pricey to do. This is a complicated multiplex PCR assay. It's really state of the art. The other thing is you either have a choice to adopt it and use it in a defined patient population or not. It's not like you can ask on a patient by patient basis whether you should use the test, because you're really, burning time if you're going to wait to decide who you're testing. So we had a lot of questions when these first came out. This is what was on the original BioFire BCID panel. Now it's expanded to BCID2 panel, but this is the panel we were looking at, and we thought, well, how can we really answer the question as to whether this adds value in our clinical practice? And the answer was a randomized controlled clinical trial. And so we executed this trial almost immediately after this panel was approved by the US FDA, because we wanted to use it, but we wanted to make sure we used it only if it added value. And so this question was answered through this clinical trial. We took patients with positive blood culture bottles, and in the laboratory, we did a stratified randomization based on age, ICU status, and whether they were on a transplant service to one of 3 arms. Now, you might wonder, well, you just talked about 2 things, but what's the third? So the first, shown in green here, is our standard of care, where we're doing our gram stain and our subculture and susceptibility. And then in the middle, in blue or purple, is the rapid test. So we add that on to our standard of care. And then the third arm, which is in red on the right-hand side, is where we ran the rapid test, but we also involved a stewardship team. And the stewardship team was notified immediately of that result and took a look at the patient's medical record in the context of what was going on with the patient, what this result showed, and if they thought that there should be a therapy adjustment, escalation or de-escalation, then they picked up the phone and called the service and made that recommendation. And in this study, this was done pretty much 24/7. So it was a pretty involved study. So what did we find? Well, you might hope, I guess, that you would save lives. So, or you would get patients out of the hospital. But when we looked at clinical outcomes of length of stay, 30-day mortality, 30-day readmission with the same organism, toxicity, adverse drug reactions, blood culture clearance within 3 days, or acquisition of C. diff or drug-resistant organisms, we did not see any differences. And you might say, well, okay, there's no value of the test then in clinical practice, at least in routine use, as you used it in this study. But we looked at one other outcome, and I think this is really important to think about, and this is antibiotic use, specifically antibiotic modifications. So here I'm showing you the subgroup of patients who had on-panel organisms, and you see this 3 groups arranged vertically: control, the rapid test, and the rapid test, plus the stewardship intervention. And what you see is the panel did identify the organism that was there very quickly. We ran this test 24/7. It's an hour test, and we got those results out very, very quickly. You also see an E in a triangle. That's escalation of antibiotics. And you see that that happened very quickly with the rapid test, whether or not there was stewardship involvement. So if we told our clinicians that there was MRSA in the patient's blood and the patient was not on an MRSA antibiotic, they would escalate to that. That was pretty straightforward. But you also see with the D and the upside down triangle, that's de-escalation. The de-escalation happened faster only in the context of the rapid test plus antibiotic stewardship. And that was very important to us, because what happened was this was so fast that our clinicians may be notified in the middle of the night when someone else is covering, or seemingly too early to have a result, do I trust it, really questioned whether they should de-escalate at that early time point, and needed the nudging of the stewardship team to move. So overall, the finding of this study was that this rapid multiplex PCR panel led to more judicious antibiotic use without worsening clinical outcomes. And I do think that's an important outcome here. Sometimes if you go too fast, things can not go so well, but that was fine. It also led to more narrow spectrum use of antibiotics for gram-positive organisms, decreased use of piperacillin and tazobactam, importantly, decreased treatment of contaminants, because a fairly significant percentage of positive blood cultures are contaminants, and this gets to that answer very quickly. And then increased and faster de-escalation and escalation of antibiotics. But as mentioned, de-escalation was only faster in the context of the rapid test plus stewardship. And the other thing we learned is that there wasn't a lot of information about gram-negative bacteria coming from this panel. Yes, it identified some gram-negative bacteria, but the only resistance marker was KPC, and we didn't have any KPCs in this study. So our clinicians, even though we told them, for example, there was E. coli in the patient's blood a little earlier, didn't really have the information they needed to move on and change the antibiotics. So more on that later. So this is how we reported our results. Now I'm showing you current result reporting with the BCID2, and I think this is important as well. So on the left-hand side is what we see in the laboratory from these tests. So you'll see a number of positive signs. Those are the things that are positive on the assay. But what you see in the middle column and over to the right-hand side in red and in green, respectively, that's what we report out. And we try to put in comments in our report that tell our clinicians what it means that we found all these things on a molecular test. So for example, the top example is an example of Staphylococcus aureus complex. MECA is detected, so we say that, but then we explain what that is, that it's methicillin or oxacillin-resistant Staphylococcus aureus, or MRSA. And we also go on to say that MRSA is predictably resistant to beta-lactam antibiotics except for ceftaroline, and that vancomycin or another anti-MRSA treatment is recommended for initial therapy pending susceptibility results. And on the bottom, you see an example of Staphylococcus epidermidis, and here MECA is detected as well. And we call out methicillin or oxacillin-resistant Staphylococcus epidermidis, but we also mention that this is a possible blood culture contaminant unless it's isolated from more than one blood culture draw or the clinical case suggests pathogenicity. And we say, I know this is obvious, but we think it really does help that no antibiotic treatment is indicated for blood culture contaminants. So as I mentioned, one of the challenges with this study that we did with the BCID panel was there wasn't so much value on the gram-negative bacilli side. So then we turned to another technology that came out that uses cell imaging to do rapid phenotypic susceptibility testing of organisms that have grown in positive blood culture bottle. This is the Accelerate Pheno system, and it takes the contents of the blood culture bottle, it purifies out the bacteria, and then it identifies them using fish probes, and then does real-time imaging of cells in the context of various different antibiotics and concentrations thereof to get to rapid phenotypic susceptibility. In about 8 hours, it's all automated, so it's within an instrument, but like the BCID panel, this is a very expensive technology. And so this is just some examples of what goes on inside the instrument. The blood culture bottle contents are loaded, and then the bacteria are separated away from the contents of the blood culture bottle by gel electrofiltration, and then they're electrochemically concentrated and moved into wells with different concentrations of various antibiotics and watched by a camera that interprets what's happening and translates that into phenotypic susceptibility. It's all within the instrument, so it's not something that we in the laboratory have to look at and interpret, which is good. So it can be deployed and done 24/7 in a laboratory. What you see here on the top is E. coli with 4 micrograms per ml of piperacillin tazobactam. And you see on the top that the cells are turning into these long filaments. They're susceptible to the piperacillin tazobactam. On the bottom, however, the cells are growing into full colonies, and that's a resistant organism at this concentration. So really interesting technology, but again, really expensive. So when this came out, we also asked the question, is this of value in our clinical practice? And so we executed now a second randomized trial evaluating the clinical impact of this rapid phenotypic susceptibility testing method for patients with gram-negative bacteremia. This was a prospective randomized controlled trial done in 2 US sites, and it was just recently published in Clinical Infectious Diseases. And we evaluated antibiotic use and then other outcomes that I'll show you in a minute in patients with gram-negative bacteremia. And we only had 2 arms in this study because we had already learned that antimicrobial stewardship is a good thing, and we put it into both arms of this clinical trial. So we were really just comparing rapid phenotypic susceptibility testing. We're no longer looking at whether stewardship is valuable. This is what happened in this study. We started off with 1,545 subjects with possible gram-negative bacilli in their blood culture. And in the end, you can see that we end up randomizing 500, and we end up with 226 in the modified intention to treat population for standard of care and 222 with the rapid phenotypic susceptibility test. So it looks like a clinical trial of a drug, but it's a clinical trial of a diagnostic. And it's also really fun for people working in the laboratory to do this kind of studies because the randomization itself happens within the laboratory. And so it gives laboratorians a chance to participate in this kind of project. The primary outcome in this study was time to first antibiotic change because we had learned from the BCID study that we were going to be less likely to see all the things that we moved to secondary outcomes, which included the 30-day mortality, length of stay, duration of broad spectrum gram-negative antibiotic use in the first 72 hours, time to antibiotic de-escalation, escalation, and hospital onset C. diff or MDRO. So when we looked at the outcomes of mortality and length of stay, readmission within 30 days, ICU stay, acquisition of drug-resistant organisms or C. diff, these were no different between the two arms of the study, the standard of care and then the rapid study, the rapid susceptibility arm. But where we did see a difference was in antibiotic changes. So antibiotic changes were faster with rapid susceptibility testing. So in this graph, you just see the time to antibiotic change. This is any antibiotic change, and the rapid test is in blue. The standard of care is in red. So you can see visually that that's faster. And then if we take a look at gram-negative antibiotic changes, because after all, this is a gram-negative study. These patients at enrollment were known to have gram-negative bacteremia. You can see that that differential is even higher, that we see faster changes with the rapid phenotypic susceptibility testing. And then when we look at antibiotic escalation, we tiered that out by whether they had a resistant organism or a susceptible organism. And not surprisingly, time to antibiotic escalation was faster in those with resistant organisms, and conversely, time to antibiotic de-escalation was faster in those with susceptible organisms. That just makes sense. Those are the interventions that you're going to do. So the rapid test helps in different ways, obviously, if you have resistant versus susceptible organisms. And this is the data put a little bit of a different way. For one thing, you see that in the drug-resistant organism category, we only had 76 cases, whereas in the drug-susceptible category, we had 372. And we defined drug-resistant as resistant to or non-susceptible to third-generation cephalosporins if the organism was a member of the Enterobacterales, a carbapenem-resistant Enterobacterales, or carbapenem-resistant Pseudomonas aeruginosa. So what we saw, again, was faster escalation, with a difference of 43 hours in the drug-resistant group, and then faster de-escalation with a difference of 13 hours in the drug-susceptible group. So the conclusions of this study were that rapid phenotypic susceptibility testing, implemented with stewardship, can optimize treatment of gram-negative bacilli bloodstream infections, that antibiotic modifications occurred faster with rapid testing than standard of care, and that gram-negative antibiotic changes occurred a median of 24 hours faster. For drug-resistant organisms, antibiotic escalations occurred 43 hours faster from a median standpoint. There was a limitation of this study. It was just 2 centers, and the rate of multi-drug resistant gram-negative bacilli was low. But I can tell you that based on these 2 studies, the BCID study and this study, which we call Hsien, we implemented both of these technologies in our laboratory, but in certain patient populations. So the BCID we use for gram-positive and we use for yeast. The Accelerate Pheno that does this rapid phenotypic susceptibility, we do for gram-negatives, and we always apply these rapid diagnostics with stewardship. So I'm going to flip to move from PCR to sequencing-based diagnostics because the BCID study was a long time ago now when I think about it, and we have a lot of new technologies coming in, and we ask this question again and again: is this a valuable test to be offering, and how can I maximize the value of this test? So I'm going to talk first about 16S ribosomal RNA gene PCR. This is a PCR that you could do on a conserved gene in all bacteria, and then you can sequence it using Sanger sequencing or next generation sequencing to determine which bacterium is there. And you can do this right out of clinical specimens. And we've been doing this in our clinical practice since 2017. So we did a look back of the patients we've tested with this kind of assay. We run it on normally sterile body fluids and normally sterile tissues. So it's a wide variety of specimen types. We looked back over our testing for about 2 years. We looked at 566 specimens from 460 patients. And this is what we found. 17% of the tests were positive overall. 90% of the positive results were associated with clinical infections. In 5% of the patients, this test impacted clinical care. And in 22% of the positive specimens, the 16S ribosomal RNA gene was clearly detected, but I couldn't resolve it with Sanger sequencing, probably because it's a mixture. So we have now moved on to use next generation sequencing here, but that was a limitation of this study. We also saw that if there was a positive gram stain in the specimen, there's a 12-fold increased odds of positivity of this kind of testing, and that cardiovascular specimens had the highest positivity rate of 37% versus the other specimen types. Interestingly, Madiha Fida, who is the infectious disease doc who led this study, also looked at how bacterial cultures performed in this patient population, which I think was an interesting way to look at it. And what she found is that 20% of bacterial cultures were positive in this subset of patients, that 87% of those positive culture results were associated with clinical infections, and that musculoskeletal specimens had the highest rates of positivity. She also found that the 16S ribosomal RNA gene PCR and sequencing approach found a probable pathogen in 10% of culture negative specimens. There was concordance between the PCR and culture in 77% of cases. And at the bottom of the slide is a summary of the sensitivity and specificity of the two approaches. And you see that they're fairly similar, but we felt that there were certain patient populations, again, in whom this kind of testing leads to really valuable findings. And in particular, cardiovascular infections, this was very helpful. So we came up with an algorithm, in this case for diagnosing infective endocarditis, that incorporated this kind of testing. And I'm not going to go over every piece on here. It obviously starts with blood cultures, and if those are negative, it goes on to Coxiella or Burnettii serology and Bartonella serology. And if those are negative and the patient comes to surgery, it goes to histopathologic evaluation of the valve. But then the test of choice after that is 16S ribosomal RNA gene PCR and sequencing. And this study was really helpful to inform this kind of testing, because we at Mayo Clinic see a lot of cases of infective endocarditis, and this can be a challenging diagnosis to make, and this technology is really helpful in that regard. So I'm going to finish with one other sequencing-based technology, and that is shotgun metagenomic sequencing. This is very popular or attractive or exciting in infectious diseases. It's a technique where we're not talking about just the 16S ribosomal RNA gene, we're talking everything. We sequence everything in the sample, and then we look at what we find with all the sequencing data. And we find human DNA, and we find bacterial DNA, and parasite DNA, and viral DNA, and fungal DNA. And if you take an RNA-based approach, you also find RNA viruses. So basically everything can theoretically be identified. But of course, this is very expensive and it's complicated to do. So the question might be, is this valuable in clinical practice? And I don't think we have the perfect answer to this, but we have been sending spinal fluid samples out to the University of California, San Francisco for shotgun metagenomic sequencing. So we did a look back on what was being found. And this is another way to look at value. Between December 2017 and December 2019, we sent 53 Mayo Clinic patient specimens out for this kind of testing, and then 27 which we received from other facilities that we sent. We call those the Mayo Clinic laboratory specimens. So overall, 80 specimens were involved in this particular study population. And what was interesting when we were able to look at the clinical characteristics, which was in the Mayo Clinic subset, is that the vast majority of these were either subacute or chronic central nervous system types of presentations, and that many had undergone pretty extensive evaluations elsewhere without a definitive diagnosis. So this is a very select population of patients, and this is what we found. In the Mayo Clinic patient population, we found 3 potential pathogens and 6 likely contaminants. And in the Mayo Clinic laboratory subjects, we found 2 potential pathogens and 1 likely contaminant. And the 3 potential pathogens that were identified in the Mayo Clinic patient population were a Bunya virus that was subsequently identified as Cache Valley virus by the CDC, actually after this was published, human herpes virus 7, and enterovirus D68. And then in the Mayo Clinic laboratory subjects, the 2 potential pathogens that were identified were West Nile virus and Toxoplasma gondii. So you can see you can detect a breadth of organisms, but the majority of this testing was negative. And so I think, we're still learning about where the value is with this kind of testing. It's very expensive, and we need to keep thinking about this and defining that value in infectious disease. Now, I showed you our infective endocarditis approach where we use algorithms. I want to point out that I really think testing algorithms, figuring out what tests you order in a particular disease, are really helpful. And so this is the algorithm that we use for gastroenteritis, acute gastroenteritis. And it basically calls out a whole number of patients that don't need any testing. But then when you find patients who do need testing for acute gastroenteritis, it drives right to a multiplex GI pathogen panel, because that's really state of the art. And you want to use the right test, but you don't want to overuse it. And similarly, we came up with a testing algorithm for a meningitis encephalitis panel that looks for bacteria, viruses, and Cryptococcus neoformans gattii. And in general, this test doesn't really replace much of anything. So if you have available rapid PCRs for herpes simplex virus, enterovirus, and Streptococcus pneumoniae antigen, you really don't need this panel. But if you don't have that available, or an immunocompromised patients or those who have been heavily treated with antibiotics, this panel might make sense. So I'm not going to go through all the details here, but the point is you need to decide how you're going to use diagnostics really before you start using them, and work together with the lab and clinicians to come up with ordering algorithms. So I'd like to summarize here. I think with the explosion of new diagnostic tests, we really have to define value. You can do that by performing clinical trials like the 2 that I showed you today that address the impact and define the ideal utilization of new tests. Results interpretation is really helpful. Some of these new tests are very complicated, and you need to put what they mean into the actual result coming from the laboratory. And then ordering guidance is really helpful, and that really can be based on ordering algorithms that define when you should and shouldn't order particular tests. I want to thank the NIH for supporting some of the research that was shown to you today, and the laboratory at Mayo Clinic that performed much of the testing that I showed you. Thank you very much.